Every living thing you have ever interacted with, from the moss growing on a damp rock, to the family dog, to the trillions of microscopic organisms floating in the air around you, is built from the exact same fundamental unit of currency, the cell. However, if you look under a high-powered microscope, you will quickly discover that nature has executed this blueprint in two entirely separate ways. These are prokaryotic cells and eukaryotic cells. To the untrained eye, they both look like microscopic blobs of jelly. But structurally, chemically, and evolutionarily, they represent two completely different strategies for survival. Understanding the difference between these two cell types is essentially understanding the history of life on Earth today. We are going inside both. We will unpack their anatomy piece by piece, examine the molecular machines running inside them, explore the fascinating evolutionary theory of how one actually swallowed the other, and look at how these differences affect medicine and your health today. Let's start by shrinking down and looking at the oldest, most resilient organisms on our planet, the prokaryotes. Prokaryotes are the ancient pioneers of the biological world. Arising nearly 3.8 billion years ago, they reigned supreme on Earth for over 1 billion years before any other life form appeared. Today, this group is comprised of two vast domains of life, bacteria and archaea. The defining characteristic of a prokaryote is hidden in its name. Pro means before, and karyon means kernel or nucleus. Quite literally, these are cells that exist before a nucleus. If we peel back the layers of a typical rod-shaped bacterium, you won't find a centralized control room. Instead, you find a beautifully streamlined, efficient system designed for rapid survival and reproduction. Let's analyze its structure, starting from the absolute outside and moving inward. First is the capsule. Not all prokaryotes have this, but many pathogenic bacteria do. It is a sticky, slimy outer layer made of polysaccharides that acts like a camouflage cloak, helping the cell evade the immune systems of higher organisms and preventing the cell from drying out. Directly beneath the capsule lies the cell wall. Unlike the cell walls of plants, bacterial cell walls are made of a unique molecular mesh called peptidoglycan. This rigid wall acts like a pressurized spacesuit. Because the interior of a bacterium is incredibly salty, water constantly wants to rush in via osmosis. Without this tough peptidoglycan wall, the bacterium would literally swell up and explode. Just beneath the wall is the plasma membrane, a classic phospholipid bilayer that acts as the ultimate gatekeeper, deciding exactly which nutrients can enter and what waste can leave. And now, let's step into the interior fluid, the cytoplasm. Because prokaryotes completely lack internal, membrane-bound walls, the inside of the cell is one single open concept room. Look at the center. Instead of a protective nuclear vault, the genetic blueprint, the DNA, floats freely in an irregularly shaped space called the nuclear region. This DNA is a marvel of efficiency. While your human DNA consists of linear strands, prokaryotic DNA is a single, continuous, tightly coiled circular loop. There are no excess, non-coding sequences here. It is pure, functional genetic code floating near this main chromosome. You will often find tiny, independent rings of DNA called plasmids. Think of plasmids as biological cheat codes. They contain bonus genes, such as the instructions on how to resist antibiotics. Bacteria can actively pass these plasmids to one another in a process called horizontal gene transfer, essentially sharing survival tips in real time. Finally, peppered throughout the entire cytoplasm are millions of tiny protein builders called ribosomes. In prokaryotes, these are classified as 70S ribosomes, which are slightly smaller and structurally distinct from the ones found in your own cells. This structural difference is critical, as we will see later when we discuss antibiotics. To move through their world, many prokaryotes are equipped with flagella, long, whip-like tails that spin like boat propellers, powered by an incredibly complex molecular motor embedded in the cell membrane. They also feature hair-like extensions called pili and fimbriae, which allow them to anchor themselves firmly to tissues or surfaces. Roughly 1.5 to 2 billion years ago, life took a massive, complicated turn. Evolution shifted from the single-room cabin of the prokaryote to a sprawling, high-tech metropolis, the eukaryotic cell. EU means true, so these are the cells with a true nucleus. Eukaryotes make up everything complex: animals, plants, fungi, and single-celled protists, the first thing that strikes you about a eukaryote is its sheer scale. It is massive, often 10 to 100 times larger than a bacterium. If a prokaryote is a bicycle, a eukaryote is a commercial jet airliner. To run a cell this large, you cannot just let things float around blindly. You need organization. You need compartmentalization. This is achieved through membrane-bound organelles, specialized internal rooms wrapped in their own lipid membranes, allowing completely different chemical reactions to happen simultaneously without interfering with one another. Let's tour the major structures of this cellular city. At the absolute center sits the command center, the nucleus. This is a massive spherical vault protected by a double-layered membrane called the nuclear envelope, which is studded with tightly regulated nuclear pores. Inside this vault sits the master instruction manual, the DNA. Unlike the circular loop of the prokaryote, eukaryotic DNA is linear and wrapped tightly around specialized packing proteins called histones, forming structured chromosomes. Deep inside the nucleus is a dense core called the nucleus, which has one primary job: manufacturing the components for ribosomes. Once those ribosomes leave the nucleus, they enter a sprawling industrial zone wrapping around the nuclear vault, the endoplasmic reticulum, or ER. The ER comes in two flavors. The rough ER is heavily studded with ribosomes, giving it a bumpy appearance under a microscope. These ribosomes are ATS ribosomes, larger and heavier than the prokaryotic variety. The rough ER folds and modifies newly made proteins. Directly attached to it is the smooth ER, which lacks ribosomes. The smooth ER is a chemical refinery, specializing in synthesizing lipids, constructing cell membranes, and detoxifying harmful chemicals or drugs. Once the proteins and lipids are built, they need a shipping label and a delivery van. They are transported via tiny membrane bubbles called vesicles to the Golgi apparatus. Think of the Golgi as the FedEx sorting facility of the cell. It looks like a stack of deflated pancakes. It receives raw materials, modifies them chemically, packages them into clean vesicles, and tags them with molecular destination codes so they go exactly where they are needed, either to other organelles or completely out of the cell. Now, a city this large requires an immense amount of electricity. Enter the mitochondria, the power plants of the cell. Mitochondria are distinct, bean-shaped structures that perform cellular respiration. They take the food you eat and turn it into a chemical fuel currency called ATP. If we were looking at a plant cell right now, we would also see large, bright green ovals called chloroplasts. These organelles perform photosynthesis, capturing photons of light from the sun and using that solar energy to build sugars from carbon dioxide and water. Managing waste in this city are the lysosomes and peroxisomes. These are essentially recycling centers and hazardous waste containment units filled with powerful digestive enzymes and acids. They break down worn-out organelles, destroy invading viruses, and neutralize toxic metabolic byproducts. Holding this entire bustling city together is an invisible structural framework called the cytoskeleton. Made of a dynamic network of microfilaments and microtubules, the cytoskeleton acts simultaneously as steel scaffolding to give the cell its shape, and as a complex railway system. Specialized motor proteins literally walk along these microtubule tracks, carrying heavy vesicles across the cell. Now let's summarize. A eukaryotic cell is a complex, highly organized structural unit distinguished by a membrane-bound nucleus that safely houses its genetic material, DNA. Surrounding the nucleus is the cytoplasm, a gel-like fluid that contains a variety of specialized structures called organelles, each performing distinct tasks to keep the cell alive. Energy production occurs within the mitochondria and chloroplasts in plant cells, while proteins and lipids are built, modified, and transported by the collaborative efforts of ribosomes, the endoplasmic reticulum, and the Golgi apparatus. Cellular waste is broken down by lysosomes, and the entire cell maintains its structural shape, internal organization, and movement through a supportive network of protein filaments known as the cytoskeleton. Finally, this entire internal environment is enclosed and protected by a selective plasma membrane that carefully controls which materials enter and exit the cell. With all these vast differences in complexity, size, and organization, it is easy to forget that these two cell types are fundamentally cut from the same cloth. In fact, if you strip away the organelles and the cell walls, every single living cell on this planet requires a core set of 4 matching tools to exist. This shared anatomy is proof of a single, common universal ancestor. First, both types must have DNA. Whether it is a loose circular loop in a bacterium or a tightly packed chromosome in a human liver cell, DNA is the universal language of life. It uses the exact same 4 chemical bases, adenine, thymine, cytosine, and guanine, to write the code for any organism. Second, both require plasma membranes. Every cell needs a clear boundary between self and the outside world. This barrier is universally made of a phospholipid bilayer, creating a selective shield that maintains homeostatic balance inside the cell. Third, both cells are filled with cytoplasm. This watery, gel-like substance is more than just filler fluid, it is a highly active chemical soup where salts, sugars, amino acids, and enzymes are constantly colliding to fuel metabolic reactions. Fourth, both absolutely depend on ribosomes. Life cannot function without proteins. Proteins are the structural beams, the enzymes, the gatekeepers, and the workers of life. Because both prokaryotes and eukaryotes must build proteins to survive, they both carry these incredible RNA and protein machines to translate genetic instructions into physical reality. Now we know every living thing on Earth is built from a cell. But before we look at how cells differ, we have to look at what makes them work in the first place. Whether we are looking at a primitive bacterium or a complex human cell, they share 4 fundamental ingredients. Both types rely on a selective plasma membrane to control what enters and exits. Both are filled with a gel-like cytoplasm that holds everything together. Both utilize ribosomes to manufacture vital proteins. And crucially, both use DNA as their genetic blueprint. But how they organize these identical tools creates the ultimate divide in biology. Let's start with that genetic blueprint, DNA. In our previous look at eukaryotic cells, we saw that they lock their DNA safely away inside a highly organized, double-membrane vault called the nucleus. This is the defining feature of a eukaryote. Prokaryotes, however, skip the vault entirely. Their circular DNA floats completely free in a concentrated, open-air region of the cytoplasm called the nuclid. This brings us to internal organization. Remember how eukaryotic cells use a vast network of membrane-bound organelles like mitochondria and the Golgi apparatus to compartmentalize tasks. Prokaryotes don't do that. Think of a prokaryotic cell as a single-room studio apartment, everything happens in one open space. A eukaryotic cell is a multi-room mansion. While both cells use ribosomes to build proteins, a prokaryote's ribosomes float freely in an open room, whereas a eukaryote embeds them onto specialized highways like the rough endoplasmic reticulum. Because eukaryotes are highly compartmentalized, they can grow up to 10 times larger than prokaryotes. To support this massive size, eukaryotes rely on a dense, intricate scaffolding of protein filaments called the cytoskeleton to maintain their shape and move cargo. Prokaryotes have a much simpler, rudimentary framework because their tiny, lightweight structure doesn't require heavy-duty internal support. In the end, prokaryotes and eukaryotes are striving for the exact same goals. They use the same plasma membrane to protect themselves, the same cytoplasm to host reactions, the same DNA to store data, and the same ribosomes to build life. Prokaryotes achieve this through tiny, open-concept efficiency. Eukaryotes achieve it through large, complex compartmentalization. 2 brilliant architectural designs, built from the exact same foundations. This leaves us with one of the greatest mysteries in evolutionary biology: how did we get from the simple, single-room prokaryote to the hypercomplex, organelle-filled eukaryote? For decades, scientists puzzled over this massive evolutionary leap. The answer turns out to be an ancient story of a microscopic hostile takeover turned into a permanent partnership. This is known as the endosymbiotic theory, championed heavily by biologist Lynn Margulis in the late 20th century. The theory suggests that billions of years ago, a large, primitive, single-celled organism tried to eat a small, free-living prokaryote. This small prokaryote was highly efficient at using oxygen to generate massive amounts of energy. Normal predator-prey relationships end with digestion. But in this case, something miraculous happened. The larger cell swallowed the smaller prokaryote, but instead of digesting it, it kept it alive inside. The smaller cell found itself protected from the harsh outside world with an endless supply of raw nutrients. In exchange, it shared its massive surplus of energy fuel with the host cell. This partnership was so wildly successful that the two became completely dependent on one another. Over millions of years, that swallowed prokaryote evolved into what we know today as the mitochondria. A later, separate event involving a photosynthetic bacterium created the chloroplast in plants. If this sounds like science fiction, the proof is written right inside your own body. Mitochondria are living receipts of this ancient event. They have their very own circular DNA, completely separate from the DNA in your nucleus. This mitochondrial DNA looks and behaves exactly like bacterial DNA. Furthermore, mitochondria have their own 70S ribosomes, the prokaryotic size, and they reproduce inside your cells using binary fission, the exact same division method used by modern bacteria. You literally carry the descendants of ancient bacteria inside every single cell of your body. All of this deep microscopic anatomy isn't just academic trivia. Understanding these profound structural differences between prokaryotes and eukaryotes is the entire foundation of modern medicine and pharmacology. Think about what happens when you get a severe bacterial infection, like strep throat or a urinary tract infection. Millions of pathogenic prokaryotes are multiplying inside your eukaryotic body. To cure you, a doctor prescribes an antibiotic. But think about the challenge here. How do you design a chemical weapon that wanders through your bloodstream, hunts down and kills trillions of bacterial cells, but leaves your own human cells completely unharmed? The answer lies entirely in targeting their structural differences. Take the antibiotic penicillin, for example. Penicillin specifically works by sabotaging the construction of peptidoglycan mesh walls. Human cells do not have cell walls, nor do we use peptidoglycan. Therefore, penicillin can flood your body, systematically destroying the structural armor of the invading bacteria until they rupture and die, while your own cell membranes are completely unaffected. Other major classes of antibiotics, like tetracycline or erythromycin, are designed to target and lock up the protein-making factories of the cell. Remember how prokaryotes use 70S ribosomes, while eukaryotes use 80S ribosomes. These antibiotics are precisely engineered to only fit inside the molecular shape of the smaller 70S bacterial ribosome. They jam the gears of the bacterial factories, preventing them from multiplying, while your human 80S ribosomes continue to function perfectly normal. This structural barrier is also why treating fungal infections, protozoan parasites, or cancer is so incredibly difficult. Fungi, parasites, and cancer tumors are all eukaryotic. Because their cellular structures, ribosomes, and metabolic machinery are almost identical to your own human cells, finding a chemical target that kills them without severely poisoning you is one of the hardest challenges in medical science. To wrap it all up, life on Earth is beautifully divided. Prokaryotes are the small, ancient, streamlined survivalists. They lack a nucleus, keep their circular DNA loose in the cytoplasm, use 70S ribosomes, protect themselves with rigid peptidoglycan cell walls, and continue to dominate the planet's microenvironments. Today, eukaryotes are the large, complex, heavily compartmentalized structures. They guard their linear DNA inside a double-membraned nucleus, use 80S ribosomes, harness massive energy using mitochondria, and group together to form complex multicellular networks like you and me. Yet, despite their vast structural differences, both carry the identical molecular legacy of life, a shared plasma membrane, dynamic cytoplasm, protein-building ribosomes, and the same universal DNA genetic language. Thank you so much for diving deep into cellular biology with us today. If you found this detailed architectural breakdown helpful, make sure to smash that like button, subscribe to the channel, and turn on notifications so you never miss our deep dives into the hidden world of science. Leave a comment below if you have any questions, and we will see you in the next video.